All right, folks, let's start. So we will quickly finish the part two. You have seen probably, if you have not seen the video, that's absolutely fine. It's about 25 minutes, and we will quickly go over that, and then we can. I will open the exam for you. You can review that, and whoever taken this test in the scan run, you can take your test after that. So let's finish that part. I have also posted the review, that kind of like a study guide for that, and the reason for that is that although the first part, that what we have with the cell signaling, says chapter 15 all, chapter 16 all, the material in this book particularly is too much experiment oriented and goes into the detail. It's more like a level of a graduate level course which you teach at 8420. Which is a complement of this. But as we are not going into the detail, a lot of these pictures are from the Albert's book, Molecular Cell Biology, Cell Biology, yeah. So the Albert's books, and that's the reason that you have the corresponding pictures that you need to see. I have mentioned those, and the study guide will give you the idea that which are the portion of the text that you need to read. You do not have to read it all, so that's important. I will still post the learning curve, but make sure that if you have enough time to do everything else, and on top of that you have time, do the learning curve. But again, remember that learning curve questions I cannot manipulate, so there will be a lot of questions which is outside what we are covering. So it's completely up to you. More realistic will be practicing your practice quiz, which will be up this weekend, and that's what it is. Now you have the spring break coming in, and I am the last person to tell you that you need to read throughout the spring break. I mean, like that's unrealistic. Many of you are seniors, so in here. The only thing that I can tell, that if you are in a position that you are struggling with a course, if you have some dead time, that you're just sitting in an airport, or if you're just traveling, somebody else is driving or something like that, if you have some kind of that dead time that you can utilize, try to look at the lecture videos to some extent to help yourself. But I just really do not expect that you will study every day during the spring break. That is not realistic at all. So if we just have to go in a online mode, the format will be the same. All your lecture videos will be updated, and then what I can do, that I can do the lecture part that we do in here like a Zoom optional conferencing thing. I'm not sure that whether it will technically should support 300 people at the enterprise level, but I do not expect. That's completely optional. You do not have to come to that live. Even there are 10 people, that's fine. That session, if it is really comes in, will be completely recorded. So that class lecture portion will be completely recorded and uploaded the same way. So nothing is going to change. And if it is an exam, you will take the exam online on Canvas. So really for our class, if we need to go to online, there's no issues. All right, so let's start with the material. So in the last class, we covered the initial part of this, and there are some questions that came up due to one questions, that where the amplification goes on and what are the levels of amplification. And just I want to clarify the first, that you have the receptor where your ligand is binding. So just as long as this ligand is bound to that receptor, this is on the plasma membrane. Remember that receptor is on the plasma membrane. This can activate hundreds of G protein. It is just not only one. That's the important one. So right from the first one molecule can activate hundreds of G protein. That can activate a number of adenylyl cyclase, which will produce a huge amount of cyclic AMP. So that's one of the concepts. The second one, we will although discuss this a little bit later part, that just remember that as the effect of signal transduction is different, all the cells behave a little bit different way, and their intrinsic level of any enzyme activity is different. Like a skeletal muscle will not work as an intestinal muscle, or the intestinal epithelial cells will not work as the epithelial cells of your lungs. So what can happen, that if you have a cell A and if you have a cell B, both of these cells in different areas, and this do have its own adenylate cyclase, the enzyme, and this also do have its adenyl cyclase enzyme. But the activity of these adenylate cyclase can drastically vary in between the cells. It is not the same. So what we mean by that, suppose the basal activity, now just be very careful about this term. The basal activity of this adenylate cyclase means that it has not been stimulated, not been inhibited. It is the basal activity. Suppose that is 20%. Now, when you get a stimulus, which is through a G protein stimulated, that from 20% can go to 100%. Or, yeah, I'm just saying, it can go to 100%. So this is one scenario. On the other hand, a complete different thing can happen in another different type of cell, where the basal activity of the adenylate cyclase is 100%. So without any stimulus, the adenylate cyclase is constantly pumping cAMP in that cell. And here, a GI or the G inhibitory protein comes in and makes it down to 20%. So exact opposite effect. So just try to understand that this is a cell A, this is a cell B, and in your body, this can happen. That in one cell, 20% basal activity, that can be elevated up to 100% by an activation signal, which is G stimulatory, whereas exactly the opposite can happen in the other one, which is G inhibitory. So that's basically what we are going to see and talk about. So let's take a look at the next portion. So in the next portion, what we are looking at is what is happening downstream of the G protein signaling. So when we are looking at the downstream of G protein signaling, the first part is the same. So here, what we are seeing, the ligand binds to the receptor, stimulate the GS or the G protein stimulation. Then cyclic AMP is produced, and now you have a new protein, which is protein kinase A, also known as A kinase, or sometimes it's known as just pKa. So this protein kinase A do have several subunits. You can see that there are 4 subunits, where there are 2 subunits which are the catalytic subunit, and there are 2 subunits which are the regulatory subunit. And they do have binding domains for the cyclic AMP. So once the cyclic AMP rises, they will bind to the regulatory domain, and at that time, the catalytic domain will be free from there, and now this protein kinase is active. It can do some work. So this is the downstream of what we have learned in the last class. So let's take a look that once activated by the cyclic AMP, what the protein kinase can do. So here, what we are looking at, that here you have the skeletal muscle, which again, as we said, that the effect of neurotransmitter or acetylcholine, here it will cause the contraction of the muscle. Here it is relaxation. Here it can cause a secretion. So many of these things can actually happen. And let's take a look at really what happens due to these signalings. So we are going to discuss 3 different examples, and that's pretty generalized. There are many smaller things that is beyond the scope of this class. So the first thing that what we are going to see, that how the protein kinase A regulate the glycogen breakdown in the muscle cell, one. Then we are going to look at the alteration of transcription in many different cells, and also the release of fluid across epithelia. And let's take a look at how this happens. So here, what we are looking at, that this is a pathway which we call as a cascade or relay pathway. We have talked about that relay proteins, that in step-by-step how your signal is being amplified. So this is one of the example of this, that this is the G protein cyclic AMP pathway leading to glycogen breakdown in the skeletal muscle cells. So here what we are seeing, that the first thing right here is adrenaline, and adrenaline is a hormone which is produced in the stress. Not only in the stress, but even if you are just started running or working out in a very active state, that is going to be secreted from your adrenal. So this is also known as epinephrine. it will bind to the ligand receptor complex the same way, and it will stimulate the G stimulatory protein, which again, not going into the detail, the alpha subunit would separate. It will bind to the adenylate cyclase. Adenylate cyclase will be producing a lot of these CAMP. Now, this CAMP will activate the protein kinase. So up to this we have seen. Now, see that what is protein kinase is doing here. It will again phosphorylate the next protein in the relay, which is the phosphorylase kinase. Phosphorylase kinase, again, with the help of ATP, is now phosphorylated. Now, phosphorylase kinase is now active. Now, this will again just hand over the baton to the next one, which is the glycogen phosphorylase. It will phosphorylate the glycogen phosphorylase and see that what will be the effect. So, as you know that after we take the meal, the sugars, they are converted into our glucose level rises in the blood. And then the insulin is secreted, and the insulin lowers down the glucose. The way it works, that the glucose is transported in particular cells, particularly in the liver, that will be converted into the glycogen. So it can be stored in the liver, or the muscle cells also store glycogen. That's the glycogen reserve. When you use the muscle, when it needs energy, which is in the form of ATP, this glycogen will be broken down, and it can enter into the glycolytic pathway, then Krebs cycle to electron transport chains to produce the energy. So the first part, that the glycogen conversion into the glycolytic pathway, is the conversion to glucose 1-phosphate. So you can see that the active glycogen phosphorylase is going to phosphorylate that glucose 1-phosphate once upon activation. So you can easily see ligand binding to the receptor that will stimulate hundreds of G proteins, produce a large number of the second messenger cyclic AMP, producing a large number of protein kinase A, large number of phosphorylase kinase, large number of glycogen phosphorylase. So at each step, the signal is being amplified. So in a very short period of time, you can easily see that there is a drastic change in the cellular environment. So this is one of the aspects of signaling. So this is the next one, and just a different rendition of the protein kinase A. And here also what you are seeing, that the receptor is bound to a stimulatory G protein, which is not shown. The alpha, beta, gamma subunit G proteins are not shown, but they are all here in the step 1. But final outcome of that, upon binding of the ligand to the receptor, is the activation of the adenylate cyclase, which is produce a lot of CAMP. So here, this is just a different rendition of the protein kinase A, which is the Rs are the regulatory subunit, and the CAMP binds to the regulatory subunit and lets the catalytic subunit free. So now the catalytic subunit from the cytosol is going to move into the nucleus. So what you are seeing right here, this is the double membrane nuclear membrane, and this is a nuclear pore through which the protein kinase goes into the nucleus. In the nucleus is bind to one protein, which is known as a CREB or CREB, and this is the protein which essentially will be phosphorylated by the protein kinase A. So this is again CAMP binding protein. This is upon phosphorylation, you can see that it will bind to an element which is the Cre element, and usually this Cre element, and again cyclic AMP response element, and this is usually found either on the enhancers or on the promoters of many different kind of genes. So the cyclic AMP response element will bind to this CREB protein, and that will recruit another complex of protein which is known as CBP or P300. And that will recruit, and this is basically the basal machinery or the transcription machinery, and it will lead to a lot of transcription of downstream genes. Now, the next part is not needed, but again, if you just need to just understand for reading purposes that how this actually works. So essentially, this P300 is also known as EP300. And sometimes in somewhere else, you can see that it is also called the E1A, adenovirus E1A binding protein. So how it works, that it is a histone acetyl transferase. If you remember from your gene expression, and again, remember that this part is not needed in here, just if you can correlate your previous thing. that when you need the transcription, all your DNA is wrapped tightly around the histone in the nucleosome. That DNA need to loosen up from the histone, which is called the euchromatin, so that the transcription machinery can bind, or the RNA polymerase can bind and do the transcription. To do that, the DNA need to be, the histone proteins need to be acetylated. And the acetylated histones will allow a little bit of loosen up of this DNA, and this P300 or EP300 will achieve that. It's a histone acetyl transferase that will loosen up the DNA to enhance transcription of a lot of them. So again, remember that CAMP responsive element, and right here is the C element response element binding protein that recruits the CBP, which is again the Cre binding protein, and P300 to enhance the transcription. So the next one, what we are looking at, the third thing, is the release of the fluid across epithelium. So what we are looking at here, and we are looking at the G stimulatory. If it increases the CAMP level, it will increase the pKa level, and that leads to water move from the basolateral surface to the luminal side. And this is the overview of the process, and we're going to talk about that, how the mechanism or how this actually works. And you will be able to recap a lot of material that you have learned before. So remember that from the biomembrane, we are still going back to the CFTR. So your CFTR, or the transmembrane cystic fibrosis transmembrane conductance regulator, and if you remember that, this is one of the member of the ABC family of transporters. So you have these two T transmembrane domains, and these A, those are the ATP binding domains. We just gave a kind of a simplified view of the transporter in your previous class. Here in the box, you can see that this is a little bit detailed. So here you have the N-terminal, then you have 6 transmembrane domains, which is known as MSD1. This is your membrane spanning domain 1. You have another membrane spanning domain, MSD2. And then you can see that at the end of this MSD domain, there are 2 sites right here called NBD1 and NBD2. So this is your nucleotide binding domain, the same thing as the ATP binding domain. And you have 2 of them. So NBD2 is the other ATP binding domain. The thing that we did not talk about or elaborated, we just had the picture, is this R, or the regulatory domain, which is sitting right in the middle. And this is what is important, and we are going to talk about that, how this controls the activity of the CFTR and what is the relation. And again, remember that although we talked about that it causes the problem across the lung epithelia, this is found around many of these tissues: lungs, liver, pancreas, intestine, reproductive tract, skin. Many of these epithelial layers in all of these areas do contain this CFTR transporter, or in their plasma membrane. So now let's take a look at how this is related to the signaling. So again, what we are seeing that right here, you have a G protein coupled receptor, which is activating adenylyl cyclase, producing the second messenger cyclic AMP, which in turn is activating the protein kinase A. And now this active protein kinase A is going to phosphorylate that regulatory subunit. This is absolutely essential. If this subunit, or the regulatory subunit, is not phosphorylated, the ATPs will not be able to bind to this ATP binding site, and it cannot turn on the pump. So again, I'm repeating that. Remember that we said that upon the ATP binding to the site 1 and site 2, which is NBD1 and NBD2, upon ATP binding, it will open the channel. But before ATP can open the channel, you have to have the phosphorylated regulatory subunit. After that, ATP can bind and open that channel. If this phosphorylation doesn't happen, ATP cannot bind and cannot open the channel. So that's the functioning of that. And again, remember that also we talked about that this was a confusion last time, that ABC family of transporters, it uses ATP. Although it uses ATP, remember that this is not a pump, it is a channel, because it is carrying the chloride down the concentration gradient, not up the concentration gradient. ATP is just needed to open the pump, but it is not transporting, and that's the reason that this is a transporter, it's a channel. So again, a familiar picture from your biomembrane. So what it is doing, the chloride, this is basically the airway lumen of your lungs. Think about that. And chloride is being transported into the airway lumen by the CFTR. Now, once the chloride comes in, it will attract the sodium from this area, from the basolateral surface. Sodium can move in through in between the cells. And now you have sodium chloride into the lumen along with the mucus. So now you have a salt gradient which will attract water to cross the epithelia and move in here. Remember that water cannot pass through here. You have the sporins in here, so it has to move through the epithelial cells. Sodium can pass through here because they do have these channels, but you do not have any water transporters present in the tight junctions here. So if the CFTR is defective, it will not allow the chloride to pass into the airway lumen, so it will not attract the sodium, it will not attract the water. So you have a very thick mucus, and that's the problem for your cystic fibrosis patients. So why this is relevant and where is the connection? Let's take a look at this. So here what we are looking at, that in 70% of the cases, there is a mutation at the 508 phenylalanine. And again, remember that it's just not just one. Here we are just showing just one example, but there are more than 800 mutations, but most of them involve the phenylalanine 508. So it could be any one of them, C, T, T, or any one of them. Here, just think about that we are just taking up any one of them, C, T, T, which if we just remove all of them, delete those, then you have a frame shift, and you can easily see the whole protein is going to change after this. All the amino acids are different. So here you have isoleucine, then phenylalanine, glycine, valine, and after 2-isoleucine, now you have a glycine, valine, and this is basically the delta 508 sequence. Now what happens that if you have that problem of deleting that, this will lead to a problem which is the unfolded protein response. Just due to that deletion, the protein will not fold properly. And if you remember that, it's going to be removed from the endoplasmic reticulum into the cytosol, and it's going to degrade to the protein degradation pathway. So these patients will not have CFTR in their epithelia at all. So what is the consequence for this? So let's take a look at very basic genetics, that this is an autosomal recessive inheritance. So what we mean by this, that think about that in a very simplified diagram, that you have a carrier father, which is a recessive disorder, which is a heterozygote, and you have a carrier mother, also heterozygote. So this black part is the recessive trait, which is the absence of the CFTR. So as you can see that easily, the possibilities are 25% of the population is going to be affected, which will be the homozygous for this recessive trait. So they will suffer from the cystic fibrosis problem. On the other hand, unaffected 25%. On the other hand, you can see 50% are carriers among that. And so pretty much a large population, that's what the message I'm trying to give you here, that a large population are carriers, and what it has to do with our signaling and the cell biology. Let's take a look at this. Just again, this is a slide that we have seen before. It's a recessive trait, affects mainly Caucasians. About 1 in 2,500 born in the U.S. with this disease. About 30,000 people live in the U.S. with this disease. Now, 1 in 20 individuals are carriers. That number is extremely high. I mean, like, you will just, again, argue that this is a disease condition, why this allele is so common in the population. So again, why it is so prevalent? So the scientists asked these questions, and while answering these questions, they were trying to correlate this with some disease that can be correlated with this. This gives you an idea that some different thing that many of you may have heard, this sickle cell anemia story. So the individuals who actually do have these mutations and have sickle cell anemia, they are resistant to malaria. And there is a large population of the African descent who are from the tropical and the subtropical area, who are actually, historically, they were more prone to the malaria due to the hot, humid weather, mosquito-borne. the scientists actually could correlate that probably that's the reason that this sickle cell anemia, these gene actually being selected over the period of time. And we are talking about thousands of years back. So therefore, the increased incidence of the sickle cell anemia in individual of the African descent is due to that they were resistant to malaria, did not die due to the malaria, and that's the reason for that. So can we correlate that with the cystic fibrosis? So the questions the scientists ask is that what other disease might affect this channel? And to answer that question, the first thing that what they ask is that which are the disease that turn on the G stimulatory protein, which will produce a lot of these cyclic AMP to turn on the protein kinase. So we talked about this briefly in the last class. Cholera toxin is one of those which influencing the GS. We saw this diagram in the last class. Remember that. Right here, what you were seeing that the alpha subunit is connected to the G alpha beta and the gamma subunit. And right here is the GDP bound form, which is the inactive form. Now, cholera, once it is bound to the GTP, cholera toxin locks that GTP in a permanent condition. It covalently binds to that alpha subunit and will not allow to hydrolyze that GTP, and that stimulatory effect is constantly on, means adenylyl cyclase will constantly pump out cyclic AMP. So what's the consequence? So before going to that, just a basic idea that the diarrheal disease, cholera, is caused by a Vibrio cholera, voluminous chloride secretion, which in turn will pull the sodium and pull the water out of the body. So dehydration is a huge problem, and the death occurs really due to that. And CFTR actually mediates this secretion, and we are going to see that if there is a connection in between these two. So let's take a look again that what is happening due to the cholera toxin. So as we have seen that as the G stimulatory protein locks the alpha subunit into on position, you have a higher cyclic AMP, you will have more of the protein kinase being activated, and as we have seen already that the protein kinase A phosphorylates the regulatory subunit of the CFTR. So more of the CFTR will be activated, which will leak out the chloride. Sodium will be pulled from the basolateral surface, will pull water. You have a large volume of water, extreme diarrhea, and this facilitates basically the transmission because it goes out in the environment in the water bodies, and that's probably one of the reasons that this actually happened. But what happened that the scientists did an experiment to answer the question. So the question is that is the CFTR, the high currents of that heterozygous, was selected due to the cholera. So they did an experiment in the mice. So what they did, that they knocked out the CFTR gene from the mice, and then they bred the mice so that they have homozygous, heterozygous, all in different conditions. And they exposed the mice to the cholera toxin to look what happens in the intestinal cell. So this is just, I did not specifically acknowledge this bar graphs because it is from different papers. So this is just an overview of the whole complicated thing. What they found, that you can see the y-axis, you have the intestinal accumulation of fluid where you have this phenotype. The plus is basically the normal phenotype, which doesn't have anything to do with the CFTR. So you can see that normal person who is not prone to CFTR problems, they have a high accumulation of fluid, which relates to severe diarrhea. If you have a heterozygote, the effect is a little bit less. And if you have a CFTR recessive right here, the severity of the fluid accumulation is much less. So you can easily see that in the heterozygotes and the homozygous for the CFTR, they do have an advantage if they suffered from the cholera. So maybe that historically, what we are talking about 20, 50,000 years over the period of time, that cholera may have selected this trait, and that's why we see this. So this is the correlation. Now we have another interesting thing which we are going to take a look is the inhibitory G protein in here. So we are looking at the right hand side. So inhibitory G protein, on the other hand, does exactly the opposite way. Instead of constantly turning on, it will not allow this GDP to be removed by the or replaced by the GTP. So the alpha, beta, and the gamma subunit of the G protein is constantly together and locks it in position. And we already talked about that Bordetella pertussis, or the pertussis toxin, does this. So the pertussis toxin locks the GI, or the G inhibitory proteins, off in the GDP bound state. Now the effect is the same. Now here most of the people have a little bit of difficulty. So the effect is the same as the cholera, where you have a higher cyclic AMP level, higher protein kinase A level, and higher CFTR activity. And we will again see this, like why this is a conflicting information. You've already talked about the stimulatory protein doing that, why the inhibitory protein doing that. And that's the reason I just do that. Remember that one is an intestinal epithelium, another is a lung epithelium. The basal activity of adenylyl cyclase is different in these cells. So think about that the basal activity, suppose this is now becomes your lung. This becomes your intestine, which we have seen, and now this is your lung, where you have the basal activity, just an arbitrary number. You have the basal activity of your adenylyl cyclase is 100%. So what you need to inhibit that so that it goes down. So that is not allowed by the pertussis toxin. So your basal activity of adenylyl cyclase is constantly on, so the effect is the same. It is producing a large amount of the second messenger of the cyclic AMP, producing a lot of pKa and producing the CFTR. And now basically the effect is the same. A large volume of water is discharged into the lung, and the effect of the whooping cough, which is due to the pertussis toxin, is the same. It is similar to the drowning in the water. You have so much amount of fluid accumulation inside in the lungs, there is no respiratory gas exchange. So when people drown, their lungs is filled with the water, and that's why they are asphyxiated and die. And this is the main problem. So the question is that did again this Bordetella pertussis or whooping cough selected for CFTR? The answer is that is not known, because no such studies has been done. But you can just easily see that there could be a correlation, but there is no scientific study that we know of. So that's pretty much for this chapter. And chapter means like for today, because we are going to review the exam. So we will stop here, and then we will go into the next part. So that's it. So